Hello, this is the FCI podcast. I am Attila Marton, and today we cover a very important and complex topic, and this is genetic testing of dogs and the new available testing technologies. My guest today is a molecular geneticist, research fellow at the Department of Ethology at the Eötvös Loránd University in Budapest, Hungary. Please welcome Sára Sándor. And the topic we will talk about is a relatively new and available technology, the genomic chip. And as a start, let's make something clear. This kind of chip has nothing to do with a microchip or those chips in electrical gadgets. Am I right?
Yes, basically it's a very different thing, although it is similarly some kind of technical electronical thing.
And what is it exactly, how it works? And I mean, compared to the previously known genetic tests, because genetic tests have been available for some decades now. And in case of many breeds, many breeders had to or obliged to use these tests. And why is this technology so different compared to the previous ones?
So this technology has been used for many years now in science and research to do molecular genetics and genetics testing and genetic investigations. And it has actually been introduced to dog disease detection and dog breeding strategies for a few years by now, but it needs time to get more accepted and to spread. This technology is basically based on this kind of chip, which is really not a chip, as you mentioned, because it does not contain any electronic circuit or anything. However, the instruments used to do the testing and the analysis are still require, big computer capacity and etc. So it is somehow still linked to computers and others. But the chip itself, it's called the SNP chip, which analyzes SNPs. SNPs are the small nucleotide polymorphisms, which, yes, this is a hard word in English, sorry. So these SNPs are mutations in the genome. And the chip contains several thousand of small DNA pieces. These are kind of baits linked to the chip. And when you add the DNA of the dog, of a certain dog to the chip, the DNA of the dog can bind to the similar DNA sequences which are used as baits. So for example, you have a short sequence on the chip linked to the chip. You add the DNA from the dog, and that part of the DNA can bind to this bait on the chip, because DNA, as many may know, forms a double helix. So it can bind to the other one. And therefore, if there is a mutation present at the location, at the bait, and the mutation is also present in the DNA tested, the binding will be 100%. We can say that. But if the two sequences are different, because the mutation, for example, the SNP is not there, the binding will be weaker. So the chip is based on this strength of binding. If the binding is low, the signal will be lower. And if the binding is strong, the signal will be stronger. And this is detected by the computers and by this light analysis. So this is the basic mechanism. But what is important about this technology is that you can add several 100,000 of SNPs on the same chip. And you can, by these SNPs, cover the whole genome. So these are kind of mapping points spread around the whole genome. And when you test these mapping points, you can get a blink about the constitution of the whole genome.
It sounds interesting. And can I say that just if I want to simplify the explanation, that with this technology, you can sort of scan a bigger portion of the genome of a dog, for example, not just certain mutations, but a much bigger portion, because some 100,000 is it's a huge number, I think.
Yes, actually you can cover almost the whole genome. It depends on the SNPs you test, where those SNPs are located, but usually the goal is to cover as many of the genome as possible. So this means that you have regions from the genome, spanning a few thousand, few 10,000s of these base pairs, which are the letters of the genome. And at each of these parts, you have 1 or 2 mapping points. So in a simplified way, this means that the mapping points can tell you something about the actual area where they are located.
Okay, and this is a huge amount of data, actually. And do you need to have a really high qualification in genetics to be able to read these test results? Because it is a very important thing that, okay, you have your dog tested, and what to do with the results.
Yes, if you refer to the raw results, then yeah, you have to be a highly trained professional using bioinformatics software to analyze the raw data. But after the raw data has been interpreted, so you have the results, what mutations are present and which mutations are not present, then this data can be easily understood by anyone who has basic knowledge in genetics. Maybe in some specific cases, you need some more education, for example, in some special alleles for some genes, but basically the main information can be easily understood.
And one thing is really interesting. When I saw some adverts, commercial adverts of these products, without mentioning any brand names, in many cases, the main focus was on if you want to find out if your dog is 100% purebred, or in case you have a mixed bred dog, what kind of breeds are involved in your dog. Or in some countries, if I'm right, the kennel clubs use this kind of technologies, maybe not this one, but they use DNA testing for identification of the dog, just to make sure that the parents of the dogs are valid. But besides that, there are so many other data and so many other information we can get from this technology. And I mean, for example, coat color of dogs, any other traits in the phenotype, and genetically inherited diseases as well, if I'm right.
Yes, absolutely. Because all the phenotypic parameters you mentioned, even the diseases, are somehow encoded in the genome. The extent of genetic background can be different between different phenotypes, but in many cases, there is some genetic effect that causes, for example, a disease. And these genetic effects are usually linked to certain mutations, certain changes in the genetic information. And actually, the SNP chip is something that has originally been designed to detect one-letter mutations or other short mutations in the genome. So if you know that an actual mutation is linked to, for example, a inheritable disorder, then you can add this mapping point, this mutation as a mapping point to the SNP chip. And then together with the other mapping points, you can also gain information about the actual disease-linked mutation or any kind of mutation which is linked to actual phenotype.
Okay, and currently, how many diseases are scientifically proven to have a genetic background linked to a mutation or you know
I cannot tell you an exact number, but usually the number researchers talk about and veterinarians is around a few hundred, but I think it can reach also 1,000 if we consider every possible disease which can be affected by genetic background.
And is it possible to scan for all these diseases with a genomic chip?
In theory, yes, but in practice, we are still not there because we still know very little actually about all of the genome, how it affects different diseases, especially regarding complex diseases. However, in many cases where the link between the genetic background and the disease is very strong, so for example, one exact mutation is known to cause a very specific disease, in those cases, it is rather easy to add the test for this mutation to any kind of genetic testing panel, and then search for this mutation in the investigated animals. And actually, the number of these testable mutations, testable SNPs and other mutations is increasing year by year because researchers identify new mutations linked to disease.
So it can be a very effective tool to fight genetically inherited diseases, but not all of them, because it is an interesting topic, because everyone wants to have an extra healthy dog without any kind of diseases, no illness, whatever. And sometimes I have the feeling that first of all, everything is linked to genetics. And on the other hand, people think that if a dog is genetically tested, it is a kind of 100% guarantee that the dog will have a healthy life without any illnesses. So I think this is something we need to understand and learn, that it helps you to optimize your selection, for example, but it is not something like magic wand. You can't do miracle with this. So it has its own limitations. I mean,
you can almost do miracle with this, but yes, okay. you can't. Yes.
And what kind of miracles can you do with this?
For example, selecting the optimal pairs for your dog. So it can really make a big change in the so-called quality of offspring. And I do not only refer to the disease status and health of the animals, but by identifying the SNPs and other mutations that could affect phenotype, breeders can actually start to look for matings where the phenotype can be improved. So this can also be a very effective thing, a very good goal of using this technology.
But isn't it? I think it definitely needs knowledge about
yes.
genetics for this part.
Absolutely.
But if we just concentrate on fighting genetically inherited diseases, in many countries during the last some years, or even a decade ago, breed clubs, for example, or kennel clubs started to set very strict regulations. What to do with a dog with that kind of mutation, and I also see debates between and sometimes fights between breeders when they start to argue what to do with a dog carrying a genetically inherited disease. And I know my kind of answer, that it can't be good to extract these dogs automatically from the population, from the gene pool. But what is your opinion on this, without going very deep into population genetics, because that is a huge area as well?
Yes, actually, this is an answer biologists really like to say. It depends. So it really depends on the disease and on the population where the disease is present. Because there are diseases which, for example, do not always occur when the mutation is present. So you should also consider other factors. There are diseases which, for example, occur very often when the mutation is present, but are not very, very serious. And most importantly, because dog breeds represent closed populations, these populations have their attributes like inbreeding coefficient within the population, the relativeness of the animals. This is linked to the inbreeding. And if you have a population, for example, where the animals are very close related, so the general inbreeding is high, then it's not always a good idea to automatically exclude animals with heritable diseases, especially if the disease is not very, very, very serious. Because in that case, you can lose valuable rare alleles, which would add to the still remaining genetic variability of the population. But if you have a population where the genetic variability is still high, so the inbreeding is relatively low, you can be more strict with heritable diseases. So this topic should always be discussed on a breed-specific level between, for example, breed clubs and genetics specialists.
I'm really happy you mentioned breeds and you mentioned a breed-specific approach, because sometimes I see that in many cases people try to solve dog-related matters on a very simple way. They try to find a one-size-fits-all solution, but in this case, in your opinion, what you just mentioned, you have to consider a lot of different factors to set up a strategy on the population level in a certain breed, and it does matter what kind of illness we are talking about. Because yeah, I've heard situations in certain breeds in certain countries when they started to exclude dogs from the population just to get rid of a mediumly severe illness, and then managed to fix another genetically inherited disease in the population, which were more difficult to fight. So with good intention, they managed to create a more serious situation, which is sad. So it's a bit complicated. And the other thing is, I think we need to talk about it a bit when we are talking about genetic testing, and when we talk about what to do with the test results. We also need to talk about recessive mutations, because it is a bit sad to see debates between breeders or between breeders and breed clubs when they they don't know exactly what to do with a dog carrying a mutation. Is it safe? I know the answer, but it is good to hear from a professional. Is it safe to keep a dog in your breeding management, your breeding practice, which carries a genetically inherited mutation, which is related to a genetically inherited disease?
Actually, I'm very happy that you told that you know the answer, because actually I still don't know the exact answer. It's also a question where I can say it depends. But in general, in most cases, according to our current genetic knowledge, when there is a mutation present which causes causes the disease in a homozygous form, so this is a kind of recessive mutation, most cases the animals which has only one copy of this mutation won't get the disease, so won't develop the disease. But why I mentioned that it depends, because in genetics there is always some exceptions and some specific cases, but in general, recessive mutations in heterozygous form don't really cause any problem.
Yeah, if you mate that dog with another one which does not carry that mutation, then there will be no problem in the phenotype.
Yes, in most cases, no, because then when you have a heterozygous animal, this means that the animal has one copy of the mutation and one copy of the normal gene. So this is called the for example, wild type allele. And the other animal has two wild type alleles, then the offspring cannot become homozygous for the mutation.
This is what I usually say. And I don't know if it's too complicated to talk about the exceptions, because you said it depends, and there are situations when there are exceptions and it is not too good to
yes,
use
the exceptions are usually specific cases, specific alleles, specific diseases. For example, when you have, this is not a specific thing I mentioned, just an idea that could happen. When you have a mutation which causes disease when another mutation at the other part of the genome is present. And then you cannot be 100% sure that the recessive mutation, when you have one copy of it, won't cause disease in every case. For example, if my memories are good, but sorry, I have not updated currently about this disease, but in the case of degenerative myelopathy, there's a small incidence of the disease occurring, even if the causative mutations, the linked mutations, are present in one copy. But this incidence is very small.
Okay. Yeah, I like to talk about what to do with dogs carrying recessive mutation, because if you say that, okay, my dog is affected by something bad, I don't know what it is, and you say that, okay, I exclude this dog from breeding, or the breed club says that, okay, you can't use that dog because it carries something. In that case, you in the long run, you cause more problem, I think, because if you exclude too many dogs from the population, then you will reduce the diversity of the population, if I'm right.
Absolutely.
So that's why it is important to understand how to implement these test results into the everyday life of breeding, because you will do more harm probably than good.
Yes, especially you mentioned diversity. There are already papers, so data, that when diversity, the genetic variability of the population gets lower and lower, you can get serious side effects. For example, smaller litter sizes, occurrence of more diseases, not specific genetic diseases, but for example, immune system malfunction and other stuff. So diversity in itself is also very important to be set to an optimal level.
So we can say altogether that this technology can be a really useful tool in breeding to make decisions about selection. If those two dogs will be good together, I mean genetically. But we need to understand and learn how to use the results and which part means what.
Yes.
Yes. So do you think, what is your personal, professional opinion about this? If using this technology becomes an everyday thing, because if I'm right, these tests are not really expensive, and this you have to do it once with a dog using for breeding. Do you think if it gets popular, it can help to improve dog welfare in general in the long run?
I'm pretty sure that if it gets more commonly used and people can interpret the results, which also means that it should be a community thing. So you cannot just do population management by yourself as one breeder. So breeders should really think and work in communities, and actually not only breeders, but the owners of those breeds of dogs should also be involved in this community breeding. There are actually very nice review paper about this published in 2 years, actually, so in 2019, which investigated, discussed the problem that many pet dogs who live with pet dog owners get excluded from breeding because the owners just don't want to breed them, and there are often regulations that they have to spay the dogs or neuter them, and this also causes that several dogs just get excluded from breeding for no reason. So it's not only about genetic testing. Genetic testing can be the eye. So the eye seeing what to do and what are the current state of the population, but the decision should be made on a community level.
So it will not save the world itself, but
no,
it is
itself,
a very...
no technology can save the world in itself, I think.
But if we use these tools wisely, then we can really improve things.
Yes.
And if we implement it well, if we learn how to use that, if we understand how to set up breeding strategies within a population, because as you said, it is not a one-man show. One person will not save any breed from any kind of inherited diseases. In that case, it could work. And what do you think? What it takes to get this new technology known and used and implemented into the everyday life of dog breeding?
I think it basically means education. So education is the core of everything when you want to introduce a new technology, and also to change the way of thinking. Because in dog breeding, in my personal opinion, you should develop the kind of eye, take it in the good sense, to see what's behind the phenotype. So there's not only the phenotype, but there are those many genes and alleles and everything, and they are present in the dog. And when you make a breeding decision, you actually make a decision about spreading those alleles or not spreading those alleles. So this is the knowledge behind. This is what you should see by your mind eye. So I don't know what is the English expression for that. And you can see this through the genetic testing panels.
And this is a huge help. I know you don't have a magic ball, but what do you think that the technology will develop further, or if we repeat this conversation in 5 years, 10 years, will we have more developed tools, or there will be already available genetic testing tools for the public? Because you said that the genomic chip has been used for many years, but now it started to be used for the public. So where this whole thing is heading to?
Actually, there is a continuous improvement also in the chip technology, so in these SNP chips, because year by year, new SNPs, new mutations are discovered, and they can be added to the actual set of investigated mapping points in the genome. So the SNP chips themselves improve. But regarding the global view on molecular genetics and genetic testing, the holy grail, I think, of this genetic testing and population management would be to get the whole genome data of all individuals and get a very, very super computer, which is really huge computer, and manage this big amount of data of the whole genome sequences and help to make decisions. So actually, this is a plausible future view. Because whole genome sequencing is also getting cheaper and cheaper. But yes, the generative data in that case is really huge.
Well, as I was listening to you, my memories came back about the first digital camera. And what, I don't tell you how many years ago it was when I got my first digital camera, and we were shocked to have 1 megapixel images. And now what I can do with my smartphone, it is incredible. So luckily technology develops. Okay, it has a lot of disadvantages, but luckily we have a lot of advantages, and I do hope there will be. Okay, the technical development, it is unquestionable. I do hope that the education and everything we talked about, to understand how to use the results, to understand how to implement everything into breeding strategies, etc. Hopefully, we will catch up to this technical improvement, because it is good, and it is good for everyone, I think. I do thank you for sharing your ideas about the genomic chip, because I think it is important to talk about and important to understand just the basic things. If we wanted to go into deeper into population genetics and everything, we could talk much more, I think. Much more, and maybe in the future. I thank you for being part of this episode. I do hope that everyone who listened to us could get some important parts, because it is really important to understand the possibilities, how to improve dog welfare and health. Those who hasn't followed this channel, please subscribe, and you will get notified when we air a new episode. And thank you again, Shara, and hope to see you soon.
And I also thank you for the invitation.
Thank you so much. Take care.
Take care. Bye-bye.